Hey everyone, quick biochemistry basics here. Let's talk about eukaryotic transcription. The enzyme required for the process of transcription is the RNA polymerase. The prokaryotes have only one RNA polymerase enzyme, while the eukaryotes have three RNA polymerase enzymes. RNA polymerase II is a major polymerase enzyme involved in the transcription of mRNA in eukaryotes. The RNA polymerase II, along with other proteins known as the transcription factors, are required for the initiation of transcription. The transcription factors are also known as general transcription factors. Let's see how the transcription factors are designated. The initial two letters are written as TF, which stands for transcription factor. The 2 indicates that it's a transcription factor for RNA polymerase II. The letter next to 2 can be A, B, D, E, F, and H depending on the function of transcription factor. In the first step of initiation of transcription, the transcription factor EF2D binds the Tata element in the promoter. The eukaryotic promoter is about 40 nucleotides long and located upstream and downstream of the transcription start site. About 30 nucleotides upstream to the start site, there is an AT-rich sequence known as the Tata box or the Tata element. TF2D has a protein called TBP which binds Tata sequence. TBP is also known as Tata binding protein. Once TBP binds Tata sequence, it bends the DNA by 80 degrees. This bending of DNA further helps in the binding of other transcription factors. This includes TF2A and TF2B. TF2A helps in stabilizing the binding of TF2D with the promoter. TF2B interacts with TBP and the promoter region downstream to the Tata sequence. TF2B helps in the recruitment of RNA polymerase II on the promoter. Now, the RNA polymerase cannot bind the promoter on its own. A transcription factor TF2F helps RNA polymerase II to bind the promoter. TF2F interacts with TBP and TF2B while recruiting the RNA polymerase. TF2F also prevents RNA polymerase to contact DNA outside the promoter. Next, transcription factor TF2E binds the pre-initiation complex. TF2E helps in the binding of other transcription factor TF2H. TF2H is a very large complex with total 9 subunits. Out of 9, 2 subunits have ATPase activity. Using energy from ATP, it acts like a helicase and melts the promoter. And this finally causes transition from pre-initiation complex to open complex. The remaining 7 subunits of TF2H has a kinase activity. This kinase activity phosphorylates the C-terminal domain or the tail of RNA polymerase II, leading to promoter escape and transcription elongation. Transcription elongation. Once RNA polymerase have initiated transcription, it shifts into elongation phase. The transcription factors that helps in elongation are called elongation factors. There are 2 such elongation factors, TFEB and TF2S. TFEB is recruited to polymerase by transcription activators. TFEB is a kinase protein and phosphorylates serine residues in the C-terminal domain of the polymerase. This phosphorylation stimulates elongation. The other factor involved in elongation is TF2S. Now, the rate at which the RNA polymerase transcribes the DNA is not same at all DNA sequences. At some DNA sequences, the rate of transcription is fast, while at other DNA sequences, it can be slow. TF2S helps to increase the rate of transcription at the region where the rate of transcription becomes slow. It also does not allow RNA polymerase to pause and encourages to move on. 5' capping. As the RNA polymerase starts elongation, the mRNA starts forming. The formation of mRNA occurs in 5' to 3' direction. The first RNA processing event that occurs during elongation is the 5' capping. During this process, the terminal gamma phosphate of the nucleotide is removed by the enzyme RNA triphosphatase. In the next step, guanyl transferase enzyme carries out reaction between beta phosphate of the first nucleotide and alpha phosphate of GTP. Once guanine is attached, methyl transferase enzyme attaches a methyl group to the guanine nucleotide. This structure is called 5' cap and it helps in the recruitment of mRNA on the ribosome for the initiation of translation. Termination of transcription. When the RNA polymerase reaches the end of the gene, the C-terminal domain of the RNA polymerase interacts with two proteins, CSTF and CPSF. The CSTF stands for cleavage stimulation factor and CPSF stands for cleavage and polyadenylation specificity factor. When the end of the gene is transcribed into RNA, these proteins are recruited to the mRNA by the C-terminal domain of RNA polymerase. The cleavage stimulation factor CSTF cleaves the mRNA. Once the mRNA is cleaved, CSTF dissociates. The CPSF then recruits poly-A polymerase, which adds about 200 adenine residues at the 3' end, giving rise to poly-A tail. The poly-A polymerase uses ATP for this purpose. Once poly-A tail is formed, the poly-A binding protein binds the poly-A tail, and during this step, CPSF is released from the mRNA. The poly-A binding protein prevents degradation of poly-A tail.
In eukaryotes, RNA polymerase II transcribes protein coding genes. A basal level of transcription requires a set of basal transcription factors that bind to DNA or other proteins. First, a protein recognizes and binds to the TATA sequence at the promoter. TATA binding protein, TBP, bends the DNA. Auxiliary factors bind to the TATA binding protein. This protein complex promotes binding of RNA polymerase and its associated proteins. The complete pre-initiation complex denatures the nearby DNA helix. RNA polymerase II and its associated proteins move along the unwound DNA and produce a messenger RNA complementary to the DNA template. The level of transcription is increased above the low basal levels by activators bound to specific enhancer sequences. Activators can act to stabilize the pre-initiation complex and make it easier for the TATA binding protein, or RNA polymerase, to bind to the DNA. Stabilization of the complex by activator proteins may also allow binding of subsequent RNA polymerase molecules. Activator proteins increase transcription, while inhibitor proteins decrease transcription. Together, these regulatory proteins modulate transcription by determining where, when, and how much transcription occurs in a particular type of cell.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the TATA box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the TATA box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.